Good morning. All right. In the topic of DNA repair systems, one of the first you need to remember is it can be divided into two big categories. One is direct repair, the other one is indirect repair. Direct repair refers to mechanisms in which the repair is carried out at the nucleotide, at the site of mutation itself. That's why it's called direct repair. And two examples given here is when you get pyrimidine dimers, and then the other one if one of the bases have been methylated. You'll need to remember the mechanisms. For example, you get the effect of sunlight will give you two potentially hazardous effects. For example, you get cyclobutane pyrimidine dimers and also 6,4 photoproducts. These are the two things that can happen after exposure to UV. And how repair is achieved is by the action of this photolyase. It recognizes the problem and then it attaches itself, and then with the help of visible light as a source of energy, it will repair. It will make a repair, and then in the presence of visible light, the enzyme catalyzes chemical cleavage of the dimer, restoring the normal base pairing. So that's why it's called direct repair. The other one is methylguanine. 0,6-methylguanine, what it does is it tackles issues of when the bases are methylated. What it does is it will attach itself. Alkyl guanyl alkyl transferase transfers a methyl group or ethyl group from the methyl guanine. So this is the problematic G, takes the methyl out and put it onto itself to a residue, a cysteine residue in the active site, and this results in the transferase being permanently inactivated. So it's sort of like suicide for a good cause. All right, so these are two examples for direct repair, and then for the rest you talk about indirect repair. And in these cases we see examples including nucleotide excision repair as well as base excision repair and mismatch repair. What the general process that takes place is, identify the region of interest, identify where the mutations are, cut out a chunk of DNA and remove it, and then resynthesize. So that is generally the mechanism that's involved. The difference between nucleotide excision repair and base excision repair, for example, is how the mutation is recognized in the first place. If you look at nucleotide excision repair, it involves enzyme recognizing the mutation and then making cuts at the upstream and downstream regions, and then removing this section, about 12 bases, and then resynthesizing using polymerase, and finally reattaching or sealing the gap by ligase. Right, so this is another example of it. You have the trimer, UVRAB trimer. That one recognizes the problem, and then it sort of makes a kink or exposes the region of problem. And then you have the UVRC, and then C cuts a nick, again downstream and upstream. And then there's a helicase, which is a UVRD that unwinds. And then finally you have polymerase and ligase. A little bit different for base excision repair. This is again some explanation for the nucleotide excision repair. But for base excision repair, what happens first is the removal of the damaged base, and then what you create is what they call as a basic site. That means it doesn't have a base. It can be a pyrimidinic or a purinic site, and then upon recognition by the N-glycosylase, AP endonuclease, then cuts again the problematic site, and then it removes it. It does a process called nick translation. Again, when do you see nick translation? You should have seen nick translation during recombinant DNA technology, where we talk about how do you label fragments. One of the ways to label it is by nick translation. It's not the right name to be used because it's not really translation, but what happens is that if you have nick translation, then that gap will be corrected by synthesis of a new strand, and as it synthesizes, it will remove the old strand away. So so this is new DNA. This is the nick area where it's being removed, and after doing it for several bases, then you stop and then you seal the area with ligation. Alright, so nucleotide excision and base excision repair essentially removes and then resynthesize. It's just the difference is basically how the damage is recognized and the processes involved in repairing. This one just says that if you have a problem with your repair systems, you are amenable or you are at risk of developing this disorder, xeroderma pigmentosum and Cockayne syndrome, and so on. These are some examples. Now, mismatch repair, again, normally it occurs after or during or after replication. So if there is a mismatch, so the challenge is to determine which ones to remove. Either you remove the A or remove the C, and this is determined by the presence of methyl groups in a sequence GATC sequence. How do you get this situation where you have the hemimethylate? This normally occurs right after replication. DNA strands are methylated, but when it starts making copies, you separate and then you start making copies. The original one remains methylated, but the new one is not methylated yet. So if there is a system that detects a mutation between, in this case, A and C, it will look at which strand contains the methyl groups, and that strand will be the original strand and will not be repaired. The ones that do not have the methyl groups is the new strand, and this is what happens. Once the system recognizes sequences GATC, and it recruits several proteins and binds to the region of problem and then removes, and then again similarly it will start to remove chunks of DNA and then resynthesize and then reseal the gap. Okay, and finally, why is recombinational repair important? In all of the examples that we've seen earlier, every time you have a problem, you have a template that you can use to restore the or to resynthesize as a guide to synthesize the new strand. You have a template, but in this case, you look at this problem. There is a problem here, and then there's a thymine dimer, so the repair is not possible. It doesn't have any template. So what happens here is that you will require the process of recombination, in which, and this is what happens if you have a double strand break, you use one strand as you sort of borrow one strand from your homologous chromosome, and then use that as a template for the synthesis of one, and then the other one can, because now the original double stranded it now remains a single stranded, now you can use that as a template to repair them. So now in the end, what happens in here is that you resynthesize both strands, but because they are homologous, it should be alright, because it should contain the same chromosomes. However, this later on in this course, you will see that this may also be the cause for gene conversion. Okay, the loss of if heterozygous sequences, and then the homologous chromosome may not have the same sequences, but because it's being repaired, then you lose the heterozygosity based on the template that is used for repairs. Okay, all right, see you in the next slide.